But in 1950s, they didn't think of this like this. What they saw, of course, in the cell there are like thousands of different kinds of protein and thousands of these ribosomes. And they somehow saw that these proteins are oozing from this ribosome. So what they thought, each ribosome make one protein. So each protein is different, right? Since there are thousands of different kinds of ribosome, each ribosome makes one kind of protein. So to make different kind of protein, information is somehow hidden in ribosome. So there are if there are 20,000 different kind of proteins, 20,000 different kind of ribosome. So Francis Crick, one of the great scientists of the 20th century, he hypothesized one ribosome, one protein. So message for the protein is hidden in the ribosome. But that was 1950s. Slowly by the end of 1950s, people identified mRNA. So ribosome suddenly lost all its sheen. They identified actually it's the mRNA which carries the message. Ribosome is only the factory which makes the protein based on the mRNA, right? So then suddenly the whole idea collapsed. So it is actually one ribosome which can make all 20,000 different kind of protein. So ribosome is just a factory which makes the protein. So from one protein, one ribosome can make 20,000 protein. Okay? So that was the thought almost till 2002. From 2002, people started identifying, no, it doesn't seem to be right. These ribosomes all don't seem to be same. They seems to be slightly different. If I can say use the term, they seems to have different flavors. Though there are look and do same kind of thing, they carry different flavors. If there are so many flavors, thousands of different flavors, it should mean something. So then they go back to the idea that not that every ribosome make every protein, there must be some kind of speciality. There is some kind of match happening, making of the match happening at some level, so that ribosomes are specialized to make only some kind of protein. Okay, there are some ribosomes which can make any protein, but there are specialized ribosomes, they can make only some protein. Okay, that's the concept of ribosome heterogeneity. Ribosome is not one single machine which you can make everything. It is actually a very specialized machine with very distinct flavor, and distinct flavor means something. So that's the ribosome heterogeneity. But if you would have asked me about ribosome heterogeneity some 7-8 years ago, I would have known same as you or less than you. I knew nothing about ribosome heterogeneity because that was not what I was doing. So in today next maybe in 20-25 minutes, I will take you through a story that developed in my lab, through which I learned more about ribosome heterogeneity, and hopefully I also am contributing little bit to the field, and more so I am daunted by the challenges. So actually I will take you through the story and make you realize sometimes the findings happen very serendipitously. You don't plan for, because I didn't know what is ribosome heterogeneity. I didn't plan to study ribosome heterogeneity. It just happened. So I will take you through the story because all of you are in the very beginning of research career. Hopefully you keep in mind that you can't plan everything that you want to discover or you study. The things will come to you suddenly and you just be open to look for it. Okay, my story starts with Fragile X syndrome. So what is Fragile X syndrome? So Fragile X syndrome is a neurodevelopmental disease. So our brain, when it develops, take a very specific trajectory. There is a plan, you have to go according to the plan. For this plan to completely manifest, there are so many things have to come together. So many proteins, so many RNA has to come together. If you even don't have one single protein, the whole trajectory can go wrong. So you end up not having a regular, normal, controlled brain, you will have a autistic brain. Okay? So one such protein is FMRP. This is a very, very, very common. Among the autism, this is the most common genetic cause of autism. What happened in this case is this one single gene. So if you have questions, please stop me at any time, I don't mind. Okay? So this one single gene undergoes a mutation. So this one single gene codes for one single protein. But if you don't have this one single protein, the whole brain development is defective. Okay. So what happens, the connections are problematic, the kids will have problem in learning, they have higher anxiety, they have like autistic behavior, they can't make eye connections, they have the comorbid epilepsy, all those things happen. And since this chromosome is present on X, that means like it is more prevalent in the males. So males are more likely to get it than the females. Okay. But what is it doing? So I said like it's absence of one single protein, right? What is this single protein doing? Lot of studies happened, almost like 20 years of study. After that they realized this protein controls the making of other proteins. So it regulates the protein synthesis. If you don't have this protein, you cannot have normal protein synthesis, which is required for your synapse. So you guys are all familiar with synapse. Synapse are the connection between the neurons. These are actually the fundamental units. This is where all your memory, emotion, everything is stored in synapse. Synapse is essential, right? So if you don't have FMRP, you don't have proper synapse, you end up having Fragile X syndrome. So I was interested to study what does FMRP do. So because most of the people, when you have a disease and you see that a protein is like causing, most of the people what they do, they try to study and understand what happens in the absence of like protein. That's what leads to disease, right? But for me, it was like if you go to watch Hamlet, and if you don't have the Prince of Denmark, it's like that. If you want to see and enjoy Hamlet, you have to have the Prince of Denmark all through in your play, right? So so that was my intention. So I have been doing this, what FMRP does, I am studying this more than 20 years now. One of the things we realized, FMRP play a very important role in regulating protein synthesis, it controls the cytoplasmic protein synthesis and so on. But one thing I realized more recently is, lot of FMRP is in the nucleus. So you guys are aware like cytoplasm and nucleus. A single protein has 1, 2, 3, 4, 5 distinct RNA binding. So if it has so many RNA binding domain, it must be binding to RNA like it does in the cytoplasm, it must be doing the same thing in the nucleus. That's what we thought. So what we did, this experiment. So what we did, again, we didn't go for neuron, because neuron is the end product. The beginning of the story is in the embryonic stem cells. Embryonic stem cells are the pluripotent cells which makes all of you. All of you start with this single pluripotent stem cell. Okay. So if you want to look at the development of brain, you want to look at the beginning. So we started looking at the embryonic stem cell. This is just, don't worry about the picture. This is just an embryonic stem cell picture. So we took these cells, collected the nucleus, because we only wanted to focus on nucleus, collected the nucleus, and we wanted to see what kind of RNA FMRP binds to. For that, there is an experiment called immunoprecipitation. You use an antibody against that, pull down and look for all the RNAs. Okay? So we look for all the RNAs. Based on our earlier result, we thought the most abundant class of RNA which binds to FMRP is microRNA. Okay? So if you can look in this gel, microRNA has tiny RNA. This is the band we expected. Okay? So there was a band, there was a small band, we thought okay, microRNAs are there. But very interestingly, there was a much bigger band at a much higher molecular weight. So what is this? So I was curious, more than me, my student was more curious, sir. Okay. So and I always thought, he said, oh Ravi, you should sequence this. We should know what is this. I said, come on Vishal, this is waste of money. In India, money is a big limitation. So I said, no, let's not do it. No, Ravi, we don't know what it is. This is microRNA, but we don't know what is this. We went on to sequence that, and that completely opened up a very new avenue, because we never expected what to find, and we found SNO RNAs. So nobody knew that FMRP interacts with SNO RNAs, and we realized that FMRP in this case interacting with a very new class of RNA called SNO RNA. Now I will jump to SNO RNA. Before that, you guys what is this guys throwing all different terms? What is SNO RNA? To understand what is SNO RNA, you need to step back 80 different proteins. So as it is made, it get modified and get folded, and proteins get bound. Okay. So this is the process. So how does this modification happens? How do they, because rRNA is a very big molecule, only 100 sites it get modified. How do you know where are these 100 sites? That 100 sites are identified by this small RNA called snoRNA. So these are called guide RNA. What happens is when the ribosomal RNA comes out, these guide RNAs are there, they sit on the right place, bring the enzyme, the methylation happens. So the modification happens based on the binding of the snoRNA. So now we understand why the snoRNA was very surprising for us and why it was very interesting, and that brought us to ribosome. Till then I had never studied ribosome. Then suddenly I got interested in ribosome because we found that FMRP, which controls neuron, bind to a specific class of snoRNA. So what is the meaning of this? So this is a very complex assay. We'll just try to take you through that. I told you in ribosome there are 100 places where this modification happens, right? The question is, if all 100 sites are fully modified or not. It turns out it's not. That means a given site can have it or not have it. It's like your binary code, it's 0 or 1. So, but that can happen on all the 100 sites. Okay. So we want to know then which of these sites are fully modified or which of these sites are not modified. How do you know? There is an assay called RiboMET-seq. I will not go into the detail. What you can see here is, if you see there that spots, there are 100 spots altogether here. If the spot is very close to 1, that means all ribosome, that site is completely modified. You cannot have that site without modification. Okay? But you see that in some sites, for example here, it's not fully methylated. Okay, it's only about 70%. That means what? In 70% ribosome, that part is modified. In 30% of ribosome, that part is not modified. Like that, you can identify all the spots and you can recognize which are the spots which are not modified. So this we did from the embryonic stem cell, human embryonic stem cell. Without any modification, we just look. If you look, you can see almost some 15 to 16 sites are variable. So 85 sites never vary, they are constant. So they are not interesting for us. Remaining 15 sites, sometimes they are methylated, sometimes they are not. So what do they mean? By being methylated or not, they actually are generating a code. A code which will tell certain mRNA can bind to this ribosome, certain mRNA cannot. This actually distinguishes which ribosomes make which protein. So this introduces the concept of ribosome heterogeneity. So I just introduced you ribosome heterogeneity, right? But ribosome heterogeneity, I said ribosomes have RNA, ribosomes have protein, all things. Why only modification? No, not only modification. The concept of ribosome heterogeneity can have many ways. It may be like, I told you there are 80 proteins. Not every ribosome bind to every 80 proteins. Some will bind to 75, some bind to 78, some bind to 80. That's one kind of heterogeneity. Other kind of heterogeneity, I already told you, the RNA and the extent of modification, the decoration on the RNA. Other than that, it can also have, it can bind to additional protein. So all these concepts actually introduce us to the overall concept that there is ribosome heterogeneity. Overall, what we are telling, ribosomes are not single monolithic machines, they have very distinct flavor, and distinct flavor is recognized by distinct mRNA. So there are specialized proteins. But again, please note here, in every cell there are tens of thousands of ribosomes. Among that, almost 9,000 ribosomes make everything. The remaining 1,000 ribosomes are more interesting. These 1,000 ribosomes are the one which have different flavors, and they are critical to determine where this happens, where it goes, where this cell goes, what happens to this cell. So we'll come back to this. So this is the ribosome heterogeneity concept. Okay. So now I told you ribosome heterogeneity can happen by multiple ways, but what we are interested is the 2O methylation. 2O methylation is like, is the decoration on the ribosome on 100 spots. This is the default one, which is methylated everywhere. So if it is methylated everywhere, it can recognize any mRNA and it can translate. Okay. So this is kind of a default. But then there are the special ribosome where are certain sites are not methylated. If they are not methylated, they are specialized. They can recognize the, there are other ways by which the mRNAs can recognize this code. So this is a code which will be recognized by an anticode, and that brings a special mRNA to that and leads to the translation. So this is the concept of ribosome heterogeneity with 2O methylation. So I introduce you to the concept, but now I will bring you to the challenges. Okay? So I told you that even in the normal cell, there is some almost 15-16 sites which are not fully methylated, that generates a code. And I told you that FMRP binds to this guide RNA which determines the code. So what is the role of FMRP? So to know that, we had to know what happens in the absence of FMRP. So we generated the embryonic stem cell because we want to do this experiment in the same genetic background. Like, if I take the cells from each of you, everything will be a mess because each of you have a very distinct genomic backgroround. If I want to understand a very specific protein, I have to do it only in the same genetic background. For that, we use a technique called CRISPR-Cas. We take a normal cell, use this CRISPR machinery to just knock out that one gene. Rest of the genes will be exactly same. We did that and we generated FMR knockout line. So then we did the same measurement of 2O methylation in the whole ribosome. So what we found, the sites which are not affected, like 85 sites, they are not affected even in the knockout. So they are fully methylated, either wild type or knockout. The remaining 50 sites which are different, partially methylated, they are the ones which are affected in the knockout. But you see, the light blue is the wild type, the dark blue is the knockout. What you can see, in the light blue, there are many of them are partially methylated. But once you knock out FMRP, many of them are fully methylated. That means there is a code generated. If you make it fully methylated, you can't generate the code. Code is lost. So FMRP, by titrating the SNO RNA, determine the making of code. If you don't have FMRP, there will be no code. So this is in the embryonic stem cells. So this work, so what's the idea here? So the ribosomes, while ribosomes are being made, FMRP titrates some SNO RNA, so it generates the code. So some sites are methylated, some sites are not. So when they come out, they lead to the specialized protein synthesis. If you don't have FMRP, this code generation is affected. So what we thought, since FMRP is very essential for your memory formation, cognition, this must be having a very important role in the neurons. So we thought, because in neurons, protein synthesis not only happen in the cell body, it also happen in the synapse. So we thought the mRNAs and ribosome which go to the synapse must be having a special code. So those protein synthesis is affected. That's why FMRP, in the absence of FMRP, you have the neuronal development, learning defects. So this was our idea, and we published this paper in 2019. From this point, all the data I will show you in the next 10 minutes, all unpublished data. So all this data was done in the embryonic stem cell. Okay. Now we thought, of course, it affects the memory, but to do that, we want to look at the neuron. So from the embryonic stem cell, want to know the neuron, what do we do? Because these embryonic stem cells are pluripotent. Pluripotent means from this cell you can make anything. You can make, yeah. That's fine. I don't think anybody. We have put it already on bioRxiv. So the unpublished data is put on bioRxiv. So it is already made public. So it's not published, but it's on the public domain already. There's no problem. Okay. So if you want to know what is this doing in the neuron, so we took this pluripotent cell, reprogrammed them to become neuron. So this is something like, this is how we did it. Okay. We take the embryonic stem cell, put some kind of factors to make them neuronal stem cell. From that it becomes neuron. So our idea is, when we are looking at the ribosome mark, the mark must be much higher in the neuron, because neuron is the one which is responsible for your memory. So the mark should be much higher in the neuron compared to the embryonic stem cell. So this is just the picture showing, these are the embryonic stem cells. From that you can get to the neuronal precursor cell. These are the beautiful, colorful, beautiful neurons which you can derive from. You can even make them in 3D, which will look like you are actually the mini brains. So, okay, we did all that, and at each stage we looked at the 2O methylation pattern. We took the embryonic stem cell, neuronal precursor and neuron. So, we expected maximum of this code should be present in the neuron. So, here comes the twist in the tail. Please, this is the same assay. Please look now. The blue color is the embryonic stem cell, the orange color is neuronal precursor cell, the green color is neuron. So, if everything is in the top means no methylation, and it's completely methylated, there is no code. If they are below, that generates the code. Now you see, maximum colored dots which you see below are actually in the embryonic stem cell. Once you turn them into neuronal precursor, most of them go up, and once you go to neuron, everything is gone. So it's completely opposite to what we expected. We thought the code will be highest in the neurons and lowest in the embryonic stem cell. It's exactly opposite. You have the maximum variability, maximum possibility of a code in the embryonic stem cell. As you go from embryonic stem cell to neuron, everything is lost. So this was a big twist in the tail. So this is in the wild type. So what happens in the knockout, in the absence of FMRP, because we are always interested to look what happens in the neuron, right? So this is what I had showed you already in the embryonic stem cell, right? So in the wild type, many of the dots are below, but in the knockout, many goes up, right? But what happens in the neuronal precursor and in neuron? See, in the topmost, there is lot of difference between the spots. That means if you don't have FMRP, clearly the code is disturbed. But look at the neuronal precursors and in neuron, the spots are completely merged. That means if you don't have FMRP, the problem in generating code happen only in the embryonic stem cells. It doesn't happen in neuronal precursor, it doesn't happen in neuron. So this is completely opposite to what we thought. Okay, so what does it mean? Why is this happening? So first we thought maybe the SNO-RNA, guide RNA, they are the one which are responsible for generating the code, right? Maybe they are different. We looked in the embryonic stem cell and in the neuronal precursor cell. There is no difference in the SNO-RNA. Even in the presence or absence of FMRP, there is no difference in the SNO-RNA. That means it's not the SNO-RNA, the guide RNA is there every time. What happens in the absence or presence of FMRP is, if you have FMRP, these guide RNAs bind to SNO-RNA, FMRP binds to the guide RNA and generate the code. But this process happens only in the embryonic stem cells. Once you convert the embryonic stem cells into neuronal precursor cell, that is lost, because even if you have FMRP, it doesn't bind. So obviously in the absence of FMRP, you don't have FMRP, it cannot bind, code is not generated, right? But that happens in the embryonic stem cell. From embryonic stem cell, when it comes to neuronal precursor cell, the difference between presence or absence of FMRP is lost. So FMRP is essential, but essential only in the embryonic stem cell stage, when there is cells are in the pluripotent form. That's what you can see. See, in the embryonic stem cell, FMRP binds, but when you differentiate the same cell into neuronal precursor, binding goes down. For all the SNO-RNA, binding goes down. The embryonic stem cell stage, the methylation is low. In the neuronal precursor, methylation goes up. The same pattern is also observed in the knockout, in the embryonic stem cell. That means, if you don't have FMRP, the embryonic stem cell already look like differentiated cell. So what is the implication of that? So this is what I am showing. If you have FMRP, it binds to SNO-RNA and reduces the methylation in the embryonic stem cells. If you differentiate into neuronal cell line, the binding of SNO-RNA to FMRP goes down, so methylation goes up. The same thing happens in the absence of FMRP. If you don't have FMRP, more SNO-RNA, more methylation, but this happens only in the embryonic stem cell. So we understood how code is, we identified there is a code, we understood how code is generated, but the big question is, what's the meaning of the code? Without understanding what's the meaning of the code, code is has no meaning. So that's where the problem is. So I completely confess, we really still don't know how exactly the code leads to certain manifestation. We are only trying to begin to dig little bit below the surface. We are really very, very close to the surface. We have haven't gone to the bottom of the things. So to even scratch the surface, this is what it was very, very challenging. What we did, if there is a code and if there is no code, what does it mean? The immediate readout for us is the protein synthesis, because the code controls the protein synthesis. So, but we don't know individual protein. We wanted to look at the total protein. So to do the total protein synthesis, there is a assay called FunCat. In this assay, you will only identify the newly made proteins. Okay. So now what we did, we took the embryonic stem cell and did the FunCat, and this is the FunCat signal. But in the embryonic stem cells, if you remove FMRP by knockout, suddenly the protein synthesis significantly goes up. Okay. This is what happened in the embryonic stem cell. But if you take the neuronal precursor cell and look at the protein synthesis, this is the protein synthesis, and if you don't have FMRP, this is the protein. So between this, there is no statistically significant difference. So difference exists only in the embryonic stem cells. If you differentiate, no difference. Okay. Now in the wild type, from embryonic stem cell to neuronal precursor, we use a different assay called puromycylation. I will not go into the detail. So here you see, this is the normal protein synthesis in the embryonic stem cell. Protein synthesis suddenly goes up. So the correlation we made is, if there is a code by methylation, protein synthesis reduces. If you remove the code, protein synthesis goes up. So the code is reducing the protein synthesis. That is happening both in the embryonic stem cell. If you remove the code in the knockout, protein synthesis goes up. In the wild type, if you differentiate, protein synthesis goes up. So hypomethylation, low protein synthesis; hypermethylation, high protein synthesis. So these are just give you hint of how structurally they look. These are the sites where there is a hypomethylation in the embryonic stem cells. In knockout, suddenly see many of these sites are hypermethylated. So the code is lost. So from embryonic stem cell, if you differentiate into neuronal precursor cell, again very similar change happens. So the wild type differentiated cell looks like pluripotent cell in the absence of FMRP. So what does it mean? So this is again I am saying, in the wild type, so there is a code. So lot of sites are not methylated, and this is regulated by FMRP. If you don't have FMRP, the SNO-RNA, guide RNA cannot be titrated, those sites get hypomethylated. So you lose the code. So if you lose the code, there is a increased protein synthesis. Okay. So in the normal case, from embryonic pluripotent cell, if you differentiate, again the sites get hypermethylated, you lose the code, again the protein synthesis goes up. But at this stage, there is no difference between wild type or absence of FMRP. Similarly, there is no difference at the neuronal stage between wild type and. So then what is actually happening? How we can explain this in the context of Fragile X syndrome? So what happens is, the pluripotent cells will differentiate. So they will differentiate into your lungs, heart, liver, everything. But when they differentiate into neuron, it depends when. The question is when. So you are the zygote is formed, a baby is conceived. From that day, only about from 3 months to 6 months, neurons differentiate. Every day, a certain kinds of neuron differentiate, and this goes to a specific spot in your brain. So the important point is not just how many neurons are made, when they are made. Because when they are made, immediately they make connection to their neighboring cells. Okay. If you don't control when they are made, the same number of neurons could be there, but their whole connection could be totally aberrant. That's what we think is happening, because if you don't have the code, there is increased protein synthesis, they are more likely to differentiate faster than the normal case. So if you differentiate faster, the connections you will make will be it may not make a much difference for your muscle or liver or kidney, but in case of brain, it make a huge difference, because in case of brain, it's not the number of cells, it's the exact connections it make, which happens only during development. So that is actually altered if you don't have the code, and that we think is responsible for altered neuronal circuit, because in the Fragile X patient the number of neurons is not reduced, only the connection, and we think this could be one of the reason why the connections are altered. So that's the story. Of course, the biggest question is how exactly code connect to specific protein synthesis is not known. We are really hoping to work more and identify. This is my team, and these are the funding agencies. Thank you.
We'll be taking questions, right?
Sir, thank you for the presentation. It was very nice. I have just two questions. First question is, this is actually a long time question that I had. It's like a chicken and the egg situation, because people say ribosomes are made out of proteins, but also proteins make ribosomes. So how are the first ribosomes even synthesized inside the cell?
So you are talking evolutionary in the cell.
In a cell, yeah.
Oh, okay, but that is easy question to answer, because like when the egg and sperm fertilize, the sperm fertilize the egg. Sperm doesn't carry any ribosome, but egg is a huge cell. It is still having lot of ribosomes. So those are all coming from mother. So they are there. So when they fertilize, the new mRNAs are made. You already have the whole set of ribosome to make this thing. So in that context, actually, I would say ribosome come first, right? So, but if you ask this question more evolutionarily, forget about the zygote. Very first cell, how did that come? So there actually the idea is it comes from the RNA. So RNA had the enzymatic capacity. RNA can carry the code. It also can do the enzymatic activity. So RNA can carry genetic information and replicate itself. So that is the thought, that it's the RNA which came first. But it may not be just RNA, some peptides bound to the RNA. So it's RNA protein world. So evolutionarily people think it's the RNA and RNP world which came first. Then it evolved slowly. That's why even today ribosome is mostly RNA. All the major functions of ribosome is carried out by RNA. We can remove all the proteins. Ribosome still can do the peptidyl transferase. So,
and second question is, like you are hypothesizing the probable cause of the Fragile X syndrome. Would it be possible to model it using some sort of organoid system where you have like a lot of stem cells, you're culturing them, and then you knock out FMRP and see when you're differentiating the stem cells into neurons, you can probably then assess if the synapses are forming or not?
Yeah, thank you. We actually have proposed this. We are seeking funds from our Department of Biotechnology. Hope they will give. Maybe in 5 years I can give you some answer.
Do we know how the variation in the amount of the number of codes leads to synapse formation exactly?
Absolutely no. That's where I said, we understand there is a code. We understand that is essential for your actual proper synapse formation. Only thing we know, the code means lesser protein synthesis. That means it's a controlled protein synthesis. If you remove the code, protein synthesis will suddenly increase. So I mean, if it is everything is making protein, there's no control. Controlled protein synthesis is essential for synapse formation. So losing the code means excessive protein synthesis, and that impacts. But how exactly this happens is mechanistically we don't know yet.
Okay. Thank you.
Sir, the Fragile X syndrome, is it transmitted from the mother, or how is it transmitted?
No. It's obviously it's transmitted through mother, because the X chromosome, there is a expansion of repeats that happens, and that happens in the mother, and it will be transmitted to either the male kid or female kid. But the female kid has the advantage that she will also have a X chromosome coming from the father, right? So it is to some extent less serious in females. In males, since they have only one X chromosome, it will be more serious. So that's this thing.
Yeah. Sir, let's say you have a mother which is infected, who has the
mutation,
and the father is normal. Then also it depends on which X is getting inactivated.
Yeah, if it's a boy,
Huh.
you are more likely to get it.
Huh.
If you are a girl, less likely to
No,
get
I
it,
mean,
because you
like-
will get a X chromosome from
Right.
one good X chromosome.
Is
from
that
father.
the reason why girls are like exactly
Girls are
two
more
times
protected
no, like,
against,
but I think from your data is also exactly 2 times compared.
Correct,
Yeah, 1 is to
correct,
3000 and
exactly,
1 is to
exactly. It's
6000.
very, very classical genetical this thing.
Hello, sir. Amazing presentation.
Thank you.
Sir, like because this snow RNA FMRP stuff happens mostly in the stem embryonic cells. Like I'm not a bio student, so I'm confused. Like basically that means the Fragile X syndrome develops in the womb.
Correct, correct.
Okay,
That's
that
what
was my
we
question.
think. That's, I mean, this is like, more at the hypothesis level, not
Okay.
proven this, but
Yes.
it mostly happens before you develop into a fully adult this thing,
So
before the brain is formed.
okay, so basically these are the proofs like for the hypothesis that
Correct.
it happens in the womb but not later
Correct,
on.
correct, yeah.
Okay.
I mean, we know that, like, even it's a developmental disorder. Unlike, Alzheimer's disease, which is a much later. This happens it can be seen very early in the babies itself. So, but what we are saying, it's not even baby. It happens in the, like, very early stages of the development. Yeah.
So you told that this FMRP and snow RNA stop binding when the cell differentiates. But the structures of each of these proteins don't change, right?
Good.
So
Yeah.
why does it stop binding?
Yes, very, very good question. So one reason we think is, so if you look at the, like, structure of these cells, when it's embryonic stem cell, the 90% of cell is nucleus. Only 10% of it is cytoplasm. Okay, but when you look at the neuron, for example, only 2% is nucleus, 98% is cytoplasm. But as I said, snow RNA, by the name itself, they are only present in the nucleus. So because once the cells differentiate, the size of the nucleus goes down, or the proportion of the cytoplasm to nucleus goes down. So that actually kind of removes the possibility of FMRP binding to the snow RNA, because the FMRP drift more to the cytoplasm. This is again a hypothesis. We have observed that, yeah, but this is one of the reason we think it should go down, because nucleus size goes down.
So by that logic, will the O methylation patterns, is it constantly seem to be affected or unaffected in all sort of cell precursors? Like we are now only talking about neuronal cell precursors.
Yeah.
Let us say some other cell precursors also, does it like go down?
Yes, that's the hypothesis. This is, I'm telling you, this is really at the cutting edge. We don't know. Whatever I'm telling is not proven. Like we know it in the embryonic stem cells to neuronal line, it's like that. But that's another question we want to. So we think it should be the same. The maximum code is actually in the pluripotent cell. As you become more specific, you lose the code. That's the overarching hypothesis.
Thank you for the presentation, sir. So, I had a question. Like, as you said, everything is now in a cutting edge stage. I was thinking, could our experiment be designed in a way like we muted FNRP, then we also the code was rising due to the SNO RNAs were complement to the gene itself.
Yeah.
Now, do we know the each SNRO sequence, SNO RNA sequence? So, after mutating the FNRN, that protein, then we can introduce all except one, the other SNO RNAs in the cell, and see how each point of that code is resulting in the change of the cell, and then we can understand what each point in the code means.
Yeah.
Probably.
We can, but there are, as I said, this is one of the major challenge. So forget FMRP. Let's say we didn't know FMRP do all this thing, right? You had the code and it changes. So, but in the now embryonic stem cell, you want to go and change the code. How do you change the code? So you can mutate the ribosome. So you can mutate the site where the methylation happened. But that's very, very challenging, because each cell has almost 200 copies of ribosome, gene encoding for ribosome. Generally, when you make a mutation, you have one copy of the gene, you mutate it. But in case of ribosome, there are 200 copies. It's almost impossible to mutate 200 copies, right? Even if you mutate some, others will be there. And also the SNO RNAs. For each site, there is not one SNO RNA, there are multiple SNO RNAs. There is a redundancy. This is a huge challenge to modify the code through RNA. So we are bit lucky that we have a protein which modifies. We can modify it through FMRP, but we still don't know the exact mechanism by which. So fine tuning that is very difficult. So that's one of the biggest challenge, how to address this generation of code, how to manipulate the code. It's very difficult. Yeah,
Thank you,
that's
sir.
the challenge. Yeah.
Good evening, sir. My question might be a little bit out of context, but I was curious. Are there any emerging therapeutic strategies that are aimed at sort of correcting FMRP-related dysregulation of ribosomes, particularly in the context of Fragile X syndrome, or maybe like specifically autism?
Yeah, I mean, there are two parts to that. One is diagnostic, one is therapeutic. So I can imagine more diagnostic tools, because not only in Fragile X, because many autism spectrum disorder, it's a spectrum disorder. There are many hundreds of different disorders. In most of the cases, there is a defect in the protein synthesis. So we think in majority of these cases, there must be a problem with the code. So if we have a way to identify, simple, because it's RNA, you can do always qPCR, PCR based method, which is very simple and cost effective. If we can come up with a way to identify the loss of code in a different kind of autism spectrum patients, we can identify what is going wrong. So that's diagnostically. Therapeutically, this is a very difficult way. As I told you, right, altering the code is very difficult. Only way we can manipulate is by expressing FMRP. So that's a very no-brainer. If you don't have FMRP, overexpress, that's not simple. There is a lot of issues with that. But there are aspects people are trying to see if we can controllably express FMRP, would it lead to, we are also trying to do that. At least in vitro we are trying. So I don't know whether it can be, it needs some way of gene therapy, which is not easy. Right? Yeah.
Thank you, sir.
Thank you so much, sir, for speaking. It was really interesting. I loved the talk. So we have a little token for you.
Oh, okay. Thank you very much. Thank you very much. I appreciate it. Thank you. And again, I wish, it was really I enjoyed discussing this. So I again wish all the best to all of you, and also enjoy the 3 days we are going to spend here. Okay, thank you.